Bismillahirrahmanirrahim. The nitrogenous bases comprise of purines and pyrimidines. Nucleotides are formed from nitrogenous bases, and these are the building blocks of nucleic acids, DNA and RNA. Purines and pyrimidines are nitrogen-containing heterocyclic rings. These are numbered in opposite directions. These structures, in addition to carbon, contain nitrogen atoms. Note that the larger purine molecule has a smaller name, and smaller pyrimidine molecule has a larger name. When referring to specific atoms of a nucleotide, each carbon of the sugar is numbered with a prime symbol to distinguish them from the carbons of the nitrogenous bases. The addition of a pentose sugar to a nitrogenous base through an glycosidic bond turns it into a nucleoside. If the sugar is ribose, ribonucleoside is produced. If it is deoxyribose, the deoxyribonucleoside is produced. Nucleotides are nucleosides with a phosphoryl group esterified to a hydroxyl group of the sugar. The 3' and 5' nucleotides are nucleosides with a phosphoryl group on 3' or 5' hydroxyl group of the sugar. If one phosphate group is attached, mononucleotide is formed. If two phosphates are attached, dinucleotide is formed, and attachment of 3 phosphates results in formation of trinucleotides. The 3' and 5' nucleotides are nucleosides with a phosphoryl group on the 3' or 5' hydroxyl group of the sugar. Presence of amide group attached to carbon 6 on purine ring makes it adenine, whereas oxygenate carbon 6 and an amide group at carbon 2 of the purine ring makes it guanine. Linkage of ribose sugar to the imidazole ring nitrogen of adenine converts it to its nucleoside form, the adenosine, and esterification of phosphate to the hydroxyl group at 3' or 5' position of ribose sugar converts it to its nucleotide form, AMP, if one phosphate group is esterified, dinucleotide or adenosine diphosphate if another phosphate group is ligated by acid anhydride bond, and ATP if two phosphate groups are ligated by acid anhydride bonds to phosphoryl group of the AMP. Likewise, linkage of ribose sugar to nitrogen atom at 9th position in imidazole ring of guanine converts guanine to its nucleoside form, the guanosine, and esterification of phosphate to hydroxyl group of ribose sugar converts it to its nucleotide form, GMP, or the dinucleotide form, or guanosine diphosphate if an other phosphate group is ligated by acid anhydride bond to GMP, and GTP if two phosphate groups are ligated by acid anhydride bonds to the GMP. An amide group at carbon 4 and oxygen atom at carbon 2 of the pyrimidine ring makes it the pyrimidine cytosine, and linkage of sugar to ring nitrogen of cytosine converts it to its nucleoside form, the cytidine, and esterification of a phosphate group to hydroxyl group of the ribose sugar may convert it into CMP, CDP or CTP depending upon the number of phosphates added. Oxygen atoms at carbons on position 2nd and 4th on pyrimidine ring makes it the pyrimidine uracil, and a ribose sugar linked to its ring nitrogen converts it to its nucleoside form, the uridine. Next, esterification of phosphates to hydroxyl group of its ribose sugar may convert it into uridine monophosphate or UMP, uridine diphosphate or UDP, or uridine triphosphate or UTP, depending upon the number of the phosphates added. The pyrimidine thymine contains oxygen atoms and carbons on second and fourth positions and a methyl group on fifth position. And linkage of sugar to nitrogen in position 1 converts it to its nucleoside form, thymidine. And esterification of phosphates to hydroxyl of ribose sugar may convert it into TMP, TDP, or TTP, depending upon the number of the phosphates added. The purine bases present in DNA and RNA are adenine and guanine. Out of pyrimidines, thymine and cytosine are present in DNA, whereas in RNA, cytosine is present, but thymine is replaced by uracil in RNA. Thus, cytosine is the base which is present in both DNA and RNA, whereas thymine is exclusive for DNA and uracil for RNA. Here we can see that the group attached to nitrogen at ninth position in purine ring dictates its fate. Here it is represented by X. So if purine is adenine, addition of ribose makes it adenosine, and esterification of phosphate to its attached ribose moiety makes it the nucleotide adenosine monophosphate. So that's the adenosine monophosphate. Similarly, nitrogenous base, if it is guanine, addition of ribose sugar to nitrogen at ninth position makes it nucleoside guanosine. So here we can see that the addition of sugar makes it guanosine, and esterification of phosphate group converts it to its nucleotide form, guanosine monophosphate or GMP. Now here we can see that the group attached to nitrogen at first position in pyrimidine ring dictates its fate. Here it is represented by X. If hydrogen is attached to nitrogen on carbon 1 of pyrimidine ring containing an amide group on carbon 4 and an oxygen at carbon number 2, it is cytosine. It is a bit confusing here that cytosine is the nitrogenous base, whereas guanosine and adenosine are the nucleosides. But for pyrimidine, cytosine is the base, and the addition of ribose makes it cytidine, which is nucleoside. And finally, the addition of phosphate to its attached ribose moiety makes it the nucleotide cytidine monophosphate. Similarly, the nitrogenous base containing oxygen at fourth and second position is uracil. Addition of ribose sugar to nitrogen at first position of the pyrimidine ring converts it to its nucleoside form, uridine, and esterification of phosphate group converts it to the nucleotide uridine monophosphate. Finally, the pyrimidine containing oxygen at fourth and second carbon and a methyl group at carbon 3 is thymine. Linkage of ribose sugar to pyrimidine ring nitrogen at first position converts it to its nucleoside form, thymidine, and esterification of phosphate group to 3' or 5' hydroxyl group of ribose sugar converts it to its nucleotide thymidine monophosphate. For nucleotides, it is important to realize that first phosphate group is esterified to ribose sugar, whereas the next two phosphates are ligated by acid anhydride